We're going to start Chapter 5, Cell Division, and this chapter is going to take us quite a while to get through. We're going to go through mitosis and meiosis both, and you'll need to know both of those not only for this test, but when you get to college. So let's take a look at the increase and decrease of cells. Cell division increases the number of somatic cells. This word somatic cells is another word for body cells, which means every cell in your body except for sperm and egg cells being developed. So from the time you go from a zygote, which is a fertilized egg, to an adult human, you increase in your number of body cells or somatic cells. So a zygote is a single cell, and you have trillions of cells as an adult human. We need to talk about a term called apoptosis. This is programmed cell death. This is something that your cells do to decrease number of cells, to remove unwanted tissue during development, such as the tail of a tadpole. You know that a tadpole doesn't have a tail when it becomes a frog, so those cells in the tail will go through apoptosis and disintegrate. The webbing between your fingers and toes, and there are some people that are actually born with some webbing between fingers and toes, and that's because the lysosomes didn't completely take away the cells that should have died between the fingers and toes during formation. Apoptosis is also a really important process during cancer prevention. If cells are damaged, apoptosis or programmed cell death can occur, and those cancer cells can be eliminated by your own body. Let's take a look at the cell cycle. A cycle indicates a loop, so it's an orderly set of stages that occur between the time a cell divides and the time you end up with daughter cells, and those daughter cells divide. It's broken up into phases, interphase, and then mitotic stages. So interphase is going to include G1, the S or synthesis phase, which DNA replication occurs in, and G2, which is growth 2, so growth 1 and growth 2. These are part of the cell cycle. However, interphase is not part of the mitotic stage. The mitotic stage includes mitosis, which is division of the nucleus, and cytokinesis, which is division of the cytoplasm. This is similar to the color sheet that you've worked on. So if you look at this, we have growth 1 in interphase. So here's where the cell is born, developed. Born's not a good choice of the word, but it's gone through mitosis, and you have a fresh new daughter cell right here, and it goes through a growth stage. Replication of DNA and more growth is in the S phase of interphase, and G2 is the growth and final preparations for cell division. So right here is where we start to have cell division occur after all this has taken place. Now, not every cell goes through all of this. Your brain cells, for example, stay in G1, and they never get past that. So not all cells completely go through the process of mitosis. Then the first stage of mitosis or nuclear division is prophase, late prophase, metaphase, anaphase, telophase, and then you have cytokinesis or cell cutting, and you have 2 new cells. Now, a lot of times this late prophase isn't put in there. It'll just be prophase, metaphase, anaphase, and telophase. All right, let's look at some normal cells. The cells round up and the nucleus collapses. The chromatin, this is all apoptosis. The chromatin is going to condense and the nucleus is going to fragment. You have blebs that are formed, and this is the plasma membrane blistering and forming those blebs. And then you have the cell that's going through apoptosis. So apoptosis is a series of events bringing about cell destruction. Capases are the enzymes that are responsible for these, and they're held in check by inhibitors and unleashed by internal or external signals, depending on what's going to cause the cell to die, whether it is webbing between fingers, whether it's a cancerous cell that your body gets rid of, whether it's just an older cell that needs to be dismantled. 5.2 deals with the control cycle of the cell in cancer. So the cell cycle is controlled by internal and external signals. Internal signals, the cell has followed normal sequence of stages. If it has, then that's great, it'll keep going. But if the cell did not follow a normal sequence of stages, it's going to stop a lot of times and have a problem, or it will go through apoptosis. There are also external signals, should the cell divide or not, and the body's response to that question. There are 3 checkpoints during the cell cycle. There's one in G1, one in G2, and one in mitosis. And these checkpoints are where the cell looks at it and says, have I messed up? Are things okay? If not, it's going to just go through apoptosis most times. If everything's okay, it'll go on to the next stage. Cyclins increase and decrease as the cell cycle continues. And the appropriate cyclin has to be present for the cell to go to the next stage. So if you can just remember, so it should be it should be 4, but the cyclin is what helps it keep in the cycle. So if the cyclin is there, then it can go to the next stage of the cycle. Here's the G1 checkpoint. Apoptosis is going to occur if the DNA is damaged at this point. Otherwise, the cell is going to continue to divide. The G2 checkpoint, the mitosis will occur if the DNA has replicated properly. So if this didn't go well, if the replication didn't go well, it'll shut itself down here in G2 and apoptosis will occur. And then spindle assembly checkpoint, mitosis won't continue if the chromosomes are not aligned properly. And we'll talk about that more when we talk about going through the stages of mitosis itself. Checkpoints are critical for preventing cancer development. A damaged cell should not complete mitosis, but instead should undergo apoptosis. So if the cell is damaged, it's better for it to be reabsorbed and no longer existing than to completely go through mitosis again. There are some things that can cause cancer. You've heard of carcinogens. Well, carcinogenesis is the development of cancer. So if a carcinogen causes cancer, carcinogenesis, or the beginning of cancer, is what this word is broken down into. It is a multi-stage process, and what happens is normal cell division is disrupted and mutations initiate cancer. We're going to talk about proto-oncogenes and tumor suppressor genes. Proto-oncogenes. These encode proteins that promote the cell cycle and prevent apoptosis. So they promote the cell to go ahead and divide and go through mitosis and make new cells and new cells and new cells, but stop the apoptosis, which could be beneficial for the cell that's not properly gone through the cell cycle correctly, so it doesn't have a chance to reproduce its nonsense that it's producing. The analogy for this is the gas pedal of a car. The cell continues through the cycle, like you push down on the pedal of a car. Mutate to become oncogenes, cancer-causing genes, gas pedal is always pressed down, and those are some things that will help you remember what proto-oncogenes do. So they mutate to become oncogenes, they are cancer-causing genes, and they act as if they're the gas pedal and they're always pressed down, so they always continue to go, 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 and nothing stops them. In this picture, what we have is a growth factor binding to its receptor right here, and that's on the surface of the cell. This RAS protein that's right here, it's part of a signal transduction pathway, and it's activated, and it will, due to this growth factor being here, it's activated, and it sends a signal down here to the nucleus and tells the genes to produce cell division, to promote cell division. So this pathway is signaled, and the DNA sends it out and it says, hey, it's time to divide and go through cell division. But that should only happen when the growth factor is in place in this receptor. When the RAS protein is mutated, that growth factor isn't needed to tell it to go ahead and activate it. So whether that presence is there or not, it's going to go ahead and reproduce and reproduce and reproduce, causing cancer. Now the tumor suppressor gene is a little bit different. It encodes proteins that stop the cell cycle and they promote apoptosis. So these are like the brakes of the car. They inhibit progression through the cell cycle. So the mutation causes the brakes not to work. So the tumor suppressor gene does exactly what it's supposed to do. It's supposed to suppress tumors forming. But if that brake is cut and this tumor suppressor gene can't stop that from happening, it's going to go through the cell cycle and it's going to cause cancer. So gene products do not inhibit the cell cycle like it should. And that covers this first and second section of Chapter 5, and we'll start with Section 5.3 tomorrow.